I'm just going to start by, hang on. The meeting is going to be recorded. That's why we're on Zoom. An apology to start with. I study humans, and I also study mice, and sometimes I study monkeys. And I'm sure I was invited here to tell you about some of the human work, because most of this place is human work. But the topics that we're most excited about in the lab now, the human work is a little bit behind the rodent work. And so we don't quite know for sure whether it all works in humans, although it's very suggestive. So I'm going to tell you what it's like in mice, and then I'm going to let you believe that it's the same in humans. And I might be able to prove that it is quite soon. The data look quite good, but they're not good enough to present yet. I'm going to be telling you three things in mice today. How do I make this move? Okay. I'm going to try to convince you that the prefrontal cortex, specifically the medial prefrontal cortex, has a model of the future. That something about the synaptic connections in the prefrontal cortex embed the structure of the world and the structure of the tasks that you're doing in the world. And because you've got those connections embedded in an entire representation of the future, you can do clever things like planning the future just by resolving dynamic patterns of activity in that network. Those are the kinds of ideas that I'd like you to get out of the talk today. I'm just going to unpack it a little bit more and show you a little bit more detail about what I mean before I show you the data, because I think it'll be easier to understand the data when you do later. So I'm just going to unpack what I mean by a model of the future, and I'm going to do it by reference to something that I think many of you will know already in the hippocampal system. For decades, we've known that the hippocampus has something like a model of the world. This is the response of a single cell in the hippocampus, and it responds whenever the animal's at one particular place, like at this tower here in this maze that it's running on now. This means together, the population of all the cells can be used to decode the animal's location. So here, the population would say the animal's here. And then it can step through a path like this, a plan that the animal's making, it'll go red, blue, green, purple. Each time the representation is active, it's always active for just one place at a time, but different places come out in sequence, right? So you see these nice sequences of different places when you decode the hippocampal activity. We know that it's a model of the world somehow, because these sequences don't just appear when the animal's making those trajectories in space. They also appear, for example, in sleep. Here they're called replay. And not only do they, the sequences that you've experienced happen in sleep, but other possible sequences that you've actually never experienced but might be true also happen in sleep. Something about the local synaptic connections in the hippocampus allows it to generate sensible, meaningful sequences of the outside world. It's like it's got a model of the world. What I'm going to try to convince you of today is that prefrontal cortex also has a similar model of the world, but it's different from that, insofar as, well, the hippocampus could only play one step of a plan at any one time and had to use time, the sequence, to represent the whole plan. Frontal cortex can represent the entire plan all at once, and it does that by instead of using different time points to represent different parts of the plan, it uses different neurons to represent different parts of the plan, right? That's what I'm going to try to convince you of today. And so the whole plan is sitting there all at once. And then I think the data for that are quite convincing, and we're not the only lab showing it now. And then I'm going to show you something with very unconvincing data, which is about the connectivity in that network. And then at the end, I'm going to show you some wildly speculative ideas that are very beautiful if true, but with no evidence for them at all. Okay, right. Okay, that's where we're going to go. Good. Okay. So where do we start if we're trying to look at the frontal cortex? What kinds of tasks are we interested in getting our mice to do? And this is one of my favorite descriptions of the frontal cortex by David Bader and Derek Nee, but David Bader's done lots of work on this hierarchy. And he contextualized the frontal cortex by saying, look, it just starts off by saying sensory motor, if you see this, do that. And then you get a bit further forward and you get context involved. When I'm in Edinburgh, if you see this, do that. And then when you get to the real prefrontal cortex, he talks about something called schematic control. And I would agree with almost everything in this figure, except I wish he drew it on the medial surface rather than on the lateral surface. But I think there are other distinctions there too. So we want a task in mice, which is something like a schematic control task in humans. And so we have to try to figure out what the relevant features are of a schema. And so I'm going to try to capture two of those features in the task that we designed for our mice. The first is a schema organizes extended sequences of goals. So like if I want to use my go and give a lecture schema, I have to buy a ticket, I have to go, often I have to fly. Today I took a train, and then I have to eventually come up to a podium and make sense. And so all of those things are part of the process of me going to give a lecture, and I know how to do it because I've done it many times before, but all the details have been very different. That's the second part of the scheme that I'm going to capture today. Knowledge is transferred across superficially different situations. So all the details about getting to Edinburgh, giving a talk in Edinburgh, they're different from the last time I gave a similar talk to this. I can't remember where it was, somewhere else. So I want to try to capture this thing, many goals in a row, details can be different in a mouse task. And then what brain regions am I going to be looking at? Well, here's one of my favorite schema tasks in humans. And this is just like comparing scenarios in fMRI where people are going to a restaurant versus where they're going to when they're going through an airport. And that the pattern similarity looks similar for all the airport scenes, even though all the details are different, and similar for all the restaurant scenes, even though all the restaurants are different. Same kinds of regions that we're talking about with narrative control, etc., etc. if you're trying to understand a story. So we're going to try to find a part of the mouse brain that's a bit like that. I know that's a tough ask, but we'll get as close as we can. Cool. But maybe if we could do something like that, find a bit of a mouse brain that's doing something a bit like this, then it might help us understand like what the computations and circuit mechanisms are that underlie some of these more complicated, interesting things like going through a restaurant, organizing goals in the human. I hope that's true anyway, or else my cross-species research direction isn't very good. Okay, so this is the task we're going to try to work with in mice. It's a task where animals have to go to 4 rewards in a row, A, B, C, D. Each time they get a reward, the reward moves to the next place in the sequence. So there's always one bait rewarded, always one tower that's rewarded, but they have to go like 1, 2, 3, 4, 1, 2, 3, 4, 1, 2, 3, 4, right? And they have to know how to get to the 4 things, to remember where the 4 things are. You have to remember what the sequence is. Go to the ticket office, go to the train, go to the lecture room, talk about nonsense. Right, that's the rule here, but here it's just 4 spatial locations. Here's a video of a mouse doing a task like this. They get very good at it. So here's B, C's down here, he knows where it is, there's no Q, he just has to remember where it is. Here's D, and then he knows to go back to A. This is one particular sequence that he has to do. Okay, and then there's the second feature of this mouse task, that tries to capture what a schema is like in humans, which is that knowledge is transferred across superficially different situations. And so in order to do that, what we're going to ask the mouse to do is to go into this maze many times, and each time it's still got the rule, there's going to be 4 things, but those 4 things are going to be different places every time, right? That's what it's trying to do. And then you're going to be trying to ask, is it the case that the mouse understands something of the schematic knowledge, the fact that there are 4 things, or are they just relearning from scratch each time? Okay, so here we go. This is some evidence from behavior that the mice have some abstract knowledge. What this is a video of the first time an animal's ever been in this version of the problem with the rewards in these locations, but having been in other versions of the problems many times. So maybe they can know that there are going to be 4 things, but not know where they are. And you can see that he's getting lost, right? So he doesn't know where B is. He's not moving fast between them. He has to go and find where they are. I'm just going to fast forward this a little bit because he takes ages to find D. Doesn't know where it is. But the clever trick he can do, if he's good at this, is once he's found D, he's counted to 4, he's remembered where the first one was, and he can walk straight back to the first one without ever doing that long search again. So obviously I've picked a very good trial here, and mice don't do it every time. And it's a particularly difficult behavior to study because you get 1 trial per task in your behavioral data. You just have to follow that. It's the first trial that you get. But nevertheless, across all mice, every single mouse you've ever recorded, then they will return to A from D more often than they'll return to A from C or B. And in fact, quite a lot of so this is less than 50%, quite a lot of the reason why that's as low as 50% is because mice just don't like turning round. And so they'll try and take a route which isn't quite the shortest path if it avoids turning round. And so if you account for those kinds of things, it becomes a bit more of an impressive effect size. But it's already a very significant effect. So that's some evidence, I think, that these mice know something about the schema, the abstract structure of the task that they're performing. Okay, this is like what we would call a 0-shot inference. They've never made that transition before, but they're doing it above chance each time. Okay, so I'm going to try to ask, how could the neurons solve a task like this? And I think the natural way to imagine the neurons solving a task like this is to imagine there are some neurons that know where you are in the task, right? There's some neurons that know I'm at the first one, I'm at the second one, I'm at the third one, I'm at the fourth one. Let's go back to the first one again. So you have neurons that know A, B, C, D, but then there's a new problem, which is if you know where you are in the task, in order to be able to do those inferences, you've got to be able to know where each one of those task locations is on this particular instantiation of the task. A is here, B is here, C is here for this version of the task, but in a different task, A, B, C, D will move around. And so you've got a binding problem. You have to bind these neurons to the correct spatial neurons, or else you can't solve some new task with a 0-shot inference. That's one way to imagine the brain solves the task. It keeps the abstract neurons, and when it's going around the first trial, it makes memories. It binds like this location to this part of the abstract task. Okay, this is actually very similar to how our best current models of the hippocampal system work. So here is a very pretty figure I like to use a lot from Howard Eichenbaum, but he's not the only person to think this. So here are the 2 inputs to the two predominant inputs to the hippocampus, the medial and lateral entorhinal areas. And he's like, okay, we know something about the structure of space in the medial entorhinal area, and we know something about what we're seeing in the lateral entorhinal area. And then we have some binding problem, which is like some abstraction about spatial coordinates is bound to like the thing that's there, and that lets me make a spatial memory. That's very similar to what I told you before. Some abstract knowledge of where you are in the task rather than space might be bound to the physical space in order to let you do a zero-shot inference. That's one way that you could solve the task. It's kind of like the hippocampal way. You can train models like this, and they recover hippocampal representations, even if you train them through like backpropagation with a neural network. James Whittington showed this. Okay, there are also other ways to think about how you might encode the abstract knowledge in a network of neurons. One is through separating the task from space and then binding them. But there are other people that have thought, well, let me just take a neural network with no fancy properties at all and just train it on many versions of the same task. And somehow, in the dynamics of the network, magically, it's going to embed the task structure such that the whole network will return to its original position after 4 rewards and then just magically get me back to where I was. So this is called meta learning. So you don't just learn one task, but you learn from many tasks, which allows the network to embed what's common amongst those tasks. And this type of approach will solve the same problem, but it will solve it entirely in activity. There's no need for new synaptic binding when you see a new task. The activity itself, dynamics of the activity carries the structure of the task. So there's a question then about whether the frontal cortex is more like these hippocampal models, which has an abstract representation, or whether it's more like these more dynamic activity-based models like this. And if so, nobody knows what the internal representations of these dynamic models might look like. And so that would be fun to find out as well. Okay, so that's the kind of question we're going to try to answer, and we're going to try to do it by recording from the medial prefrontal cortex, the prelimbic cortex, which is as good as I can get to the medial frontal cortex in a mouse, a human medial frontal cortex. And we're going to play a trick, which is the mice get really good at this task. And so by after they've done 20 or 30 of these tasks, they're so good at it, we can record them on 4 in a day. And so we can record from the same neurons in multiple different tasks. What's more, we can actually sort neurons across at least 2, sometimes even 4 days. And so maybe we'll get 6, 8 tasks, variants of this task with the same neurons. And so we can ask how they remap, et cetera, et cetera. That's what we're going to do. There's 2 other things to note about this setup. One is between each task, they're going to have a bit of sleep. And so maybe we can look at what the sleep correlations look like as a proxy for synaptic connectivity. And then the last thing is, of these 4 tasks they do in a day, critically, the very final one that they do is the same setup as the first one they did, just so we can try and measure the same thing twice to see if we can see how reproducible things are. Okay, great. Okay, so I'm now going to basically tell you what the cells do, and then I'm going to tell you what that means in terms of the representation. The first most notable thing about the cells is that they do this clever thing encoding where they are along the progress to each subgoal. So this neuron here is always firing a little bit before the reward. And I'm going to show you this figure here. It's always firing like about 80% of the way towards the reward. There's another neuron that fires like 10% of the way to reward, another one that fires 50% of the way to reward, and another one that fires all the way to the reward. In fact, the vast majority of the cells in the medial frontal cortex are modulated by where you are in your current goal, your current ambition. So I think that's not a bad way of thinking about the medial frontal cortex. It's keeping track of where you are in terms of where you want to be. You can just line these up and raster them up, and you can show the whole population of these cells, and they tile this space of how far you are along to reward in much the same way that place cells tile physical space, but they're not tiling physical space, they're tiling like how far you are to the reward. Okay, when you change task, so when you change from one reward to another, I'm going from A to B here, so subtask, I'm going from A to B here, B to C here, these things stretch. So they're not coding physical distance to a reward, they're coding like percentage. I'm halfway there, I'm 3/4 of the way there. That's what they're coding for. When you could change task, they carry on with the same percentage. If you're a halfway cell in task 1, you're also going to be a halfway cell in task 2. So they're mapping this physical problem into an abstract problem of how far you are along each of the steps to the reward. This is one of the most striking things I've seen in frontal cortex in terms of representations. Often if you record in frontal cortex, you find 15% of cells code for reward and 12% code for the prediction error. This representation is 80% of the cells in medial frontal cortex are controlled by how far you are along your progress to your immediate reward. But they also have other parts of their representation which are really interesting. And so you can see here that this cell does always code when you're 10% after the reward, but it does it more after B than it does after C, D, or A. So it also knows something about where you are in the overall sequence of A, B, C, D, as well as where you are in the little sequence of getting to your immediate goal, right? So that's good. And this little figure just shows you that that's not because it's a place cell, because in the different states, it's firing in different places. However, some of these cells are place cells. In fact, you can just do a description of these cells and you can get something like this. 80% of cells are coding goal progress. Of these 80%, about half have patterns like this. And of these half, about 40% are place cells and 60% are not place cells. That's a very sort of boring descriptive level, just so you've got the sort of data in your mind. Okay, so here's the first big question. If those cells code for A in one task, do they also code for A in the other task? That is, have they got this abstraction? Are they counting how far you are in the task? And the answer to that question is going to be no. So here is a cell in X, and then in the next task Y, it's still, look at this, it's still a 50% cell, but it's changed which state it cares about. And down here, it's a 50% cell, and it's changed which state it cares about. Here, and this is X prime, back to the original one, and it's perfectly remapped back to where it was. And there's something about where the rewards are, but it's not doing it in this nice abstract state representation. It's not generalizing across different tasks. Cool, okay. I can actually show you this for all the cells very easily because I can just plot the angle between this guy and this guy, which is 90 degrees here. So I do that across all my cells. And if I look for X versus Y or X versus Z, the angle is basically totally random, except for the fact that it's always in the cardinal directions. It's always in the cardinal directions because I've already told you cells maintain their local goal progress, right? So if I'm a 50% cell, I'm always going to be a 50% cell. So the angle is going to be either 90 or 180 or 270. If I look at X versus X prime, the angle is always naught. It's a very reproducible measure. So these guys are generalizing, but these guys are not generalizing. So it's not just coding where you are in the abstract task, it also knows something about where all the rewards are. Great. Okay, no, there isn't this beautiful, nice representation of the abstraction. So how else can you encode a sequence? Well, I just wanted to remind you about what I showed you at the start. One way is to code the sequence like this, A, B, C, D. That would need some abstraction bound to space in the hippocampus. This is the other way that you can do it. You can have entire populations of cells that still code for space, but they code for space now or space in a minute or space after that. And that would also solve the task so long as you play the trick, which is to connect these guys, these fourth guys, back to the first guys. If you did that, you could just pass the whole representation around this loop, and you'd end up back where you were before ago, which would solve this kind of task, right? So that's a different way to solve this task, and I'm gonna try and prove to you that it's true. So this is the same thing again, just now I put the loop on properly. So you've got some representation for where you are. These are those place cells I was trying to tell you about. But you've also got some representation of where you will be, where you will be in n steps or were n steps ago, halfway around the loop, and then some representation of where you just were. And the critical thing is all these representations need to be there at the same time, unlike what we know about the hippocampal place cells, right? This encodes my entire sequence simultaneously. Okay. There are other situations where we have this kind of representation in real world. It's called a drum sequencer in industry or a music box in kids' toys. Like the whole sequence can be read out from the pattern, instantaneous pattern of activity, the pattern of spikes on this music box, and you turn the representation round to play out the sequence. It's a similar way of thinking about how the representation might work. So I realize that you have to sort of think in high-dimensional worlds to imagine this, and so I'm trying to find analogies. Okay, cool. If you've got this kind of representation, then just to reinforce it, you've got lots of different cells that care about the same location, but at different times in the future or in the past, right? And so here's one of these loops. If I look at one of these rings, then this ring cares about this location, and these are different lags in the future or the past. So every cell has an anchor, one anchor and one task lag in this kind of representation. This is an anchor is like, where do I care about in space? And the lag is like, how far away in task time and time do I care about it? Great. If you're the human brain and you have this other abstraction I was telling you about, this goal progress, then you can do even better because you've converted those coordinates from time into like how progress. I'm 50% of the way there, I'm 70% of the way there. And so now, instead of this being like 3 seconds into the future or past, this being 10 seconds, now you can say, how far along my abstract progress am I in this situation? And that problem is solved by the lower level abstraction. Great. Okay. So if you have this, this system will generalize perfectly and solve any new task that you give it. Okay, so how am I going to test this? Well, every cell has an anchor and a lag. I'm now just going to show you another one of these cells. This is a cell, again, it's got a goal progress, so it's like most active, like about halfway between things. Here it's halfway between A and B, here between B and C, here between C and D. But this is like when I orient it according to like when it went through A, right? So this is organized by the abstract task. But instead of that, I could say, well, let's imagine it has an anchor and organize it by when it goes through that anchor, right? So I'm going to say, let me take 5 of these tasks and figure out where the best anchor is and then align the held out task on their anchor and see what happens then for when I hold out each one. And the answer looks something like this. Here's the best anchor across all tasks. You just fit it. And then now it looks like this. So this one points down here. Forget about this one for now. This one points down here. 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 So this cell's actually firing about 1.5 rewards after I go through this particular anchor. So when the animal's running around, and whenever he goes through this place, 1.5 rewards later, so he goes to a different place to get a new reward, and then half a reward after that, this cell fires, right? Yep. So it's like, because it's keeping track of where you are in this overall thing, and it says I was there 1.5 rewards ago, so I should be there 2.5 rewards in the future. That's what it's saying. Right. Okay. I was just going to tell you that in this task, the animal never went through this anchor, and so that's why it doesn't respond in this task. Okay, so I can now do that same analysis for all of the thousands of cells recorded, and I can say I can sort them. I can take 5 tasks. I can find all of the best anchors, and I can see how I can try and predict where the cell's going to fire in the held out task. And if I do that, if I instead of plotting X versus Y, if I plot Y versus predicted Y, now I can start to predict where the cell's going to respond in this held out task. I can predict where it's going to remap to in a totally new situation. So that's telling you that every cell has an anchor and a lag, but I can show it to you in a slightly more, in an easier way, which is this. I can take the pattern of activity, just plot it as a place cell map, a spatial map. This is where it fires. Here's the pattern of activity in these 3 tasks. I'm just showing you 3 out of the 6, but it looks a little bit random in these 3. It does in the other 3 as well. And I can just correlate these 3 place maps and say, are they the same? And then the answer is going to be no. I can do the same thing 2 rewards from now, and the answer is yes. So this is now aligned to where the animal is. This is aligned to where the animal will be. So this is like a place cell, but for the future, for where the animal will be in the future, right? And I can do that for all of these lags, and the only one where they line up, this is just the correlation between all these maps, the only one where they line up is 2 rewards from now in the future, right? So this animal has neurons in its brain that know where it's going to be in the future. Okay, which futures does it know about? Well, let's look at this distribution. Here are the lags that you can record, that you can measure this way, and you can see here there are a bunch of cells that have quite small lags. In fact, that's the dominant set of cells that have quite small lags. They look like place cells, but there are all these other cells that have much longer lags that look like they have these representations of the future. And together, it looks like this system which encodes the entire path. Okay, so I've tried to tell you that the neurons in frontal cortex encode in a slightly different way from how we understand the hippocampus. They encode all of the future at once. I'm now going to try to provide some slightly less convincing evidence, but more circumstantial evidence that something about the dynamics of the network embed the structure of a task. And there's 2 ways I'm going to try to do that. The first is I'm going to look at these sleep correlations, and I'm going to say, if I just look at a neuron, at pairs of neurons on the same ring that have the same anchor, and I ask, are they correlated in sleep? Do they fire in the same 100 millisecond window in sleep? And I relate that to where they are on the ring, what their lag is. And the answer is the answer is like a perfect circle, effectively, or a perfect ring. So neurons that are close in lag, that have like either 0 to 45 degrees away from each other or near 360 degrees from each other on the ring, are correlated in sleep much more than neurons that are 180 degrees away from each other on the ring. So it looks like that ring structure is embedded in the sleep correlations of the neurons. And wherever we know that to be the case, wherever it's been measured before, sleep correlations are a very good proxy for synaptic connectivity. Like where we know the synaptic connectivity and measure the sleep correlations, they seem to align very well. There's not many studies that have done that, but where they have been done, it looks good. So there's some suggestion that this task structure is embedded in the connections in the medial frontal cortex. The second thing I'm going to try and do is use parts of the ring a long way away to predict what's going to happen when it gets round the ring. And that could only really happen if you imagine if the whole thing, if the dynamics during behavior were also a ring. So the dynamics during sleep are a ring, are the dynamics during behavior a ring? And so I'm going to play this kind of game. I don't know if you noticed on that first task, but I showed you this mouse solving this problem where he had to go from here to here. And in the first trial here, he went down this way. And in the second trial, he's going to go this other way, I think, like this, right? And so the behavior is not entirely stereotypical. In this kind of model, where you have a representation of the entire future, that future includes the path they're going to take to get to the rewards. And we might be able to predict which of those two things that they're going to do. Is he going to go down here, or is he going to go here by just looking at the patterns of activity in the neurons a long time ago, way before he started that lab? Because the activity in the model is just going round a dynamic chain. And so if we look at it way before, and then it goes round the dynamic chain, and then it gets there and he turns right. So specifically, look, the reason this animal is turning right here and not here, we think is because there are some neurons at the top of this loop that are saying, right, that the orange path is better than the red path, or the other way around. Right? The yellow path is better than the red path, I go down. The red path is better than the yellow path, I go right, okay? But for it to be there, it had to start down here ages ago, like 20 seconds ago, or here 40 seconds ago, and then it has to pass itself around the dynamics of this loop, right? And so the question is, can we take this activity from down here and make a guess, which is like ages ago, make a guess at which they're going to do like 30 seconds in the future? And the answer is, you can do that with linear regression. You just ask, I just put the activity of these neurons into a regression and say, can they predict which choice it's going to make? And you do that. The answer is yes for the activity now, but also yes for the bottom of the loop, the activity miles ago, more than 2 rewards ago. I can predict also here, I can predict, like, look, this bump is going to carry on going around here, and in 20 seconds time, it's going to make this choice rather than this other choice, right? That can only happen if this representation is a loop, right? And so the dynamics of the representation are a loop in behavior and also in sleep. How much longer do I have? 10 minutes, great. I didn't think I'd get to the end of this talk, but I think now I'm going to. The next section of this talk, I haven't got a lot of details, and I worry that it might not take 10 minutes now. I didn't think this was going to be a problem. The next section of this talk is entirely speculative. It's a model that says if those two things are true, if I've got a model of the future in my frontal cortex, and if the world and task structures are sitting in the synapses of that model, then it would be super great because it would let us do some really cool things. And so it's trying to show you the kinds of things that you could do with such a model. And so it's going to, I think it's going to say that you can, what I'm going to try and say is that you're going to be able to infer all sorts of complicated behaviors using this representation of the future. And this is a model by Chris Jensen. The previous work you've seen so far was by Mohammed El-Gabi and Adam Harris. So this is a way to think about this model. I'm not going to show you the details of it, but I'm going to say it makes the following argument. The frontal cortex plans in a similar kind of way to how the visual cortex sees. So here's kind of a very simplistic view of visual cortex that I'm sure all the cognitive neuroscientists will disagree with. But I think we think the visual cortex is doing inference over an internal model of the statistics of the visual scene. So, and that inference is because the visual scene doesn't automatically tell you what's there, what's causing the visual scene. Instead, you have to infer what is causing the sensory stuff to happen. So the best examples of these from single unit work are like this. Here's you inferring that there's a square there, even though there's no square there. And then I can look at the cells that the cells that fire for the horizontal bar here. And even though there's no horizontal bar there, they're going to fire because the visual cortex is inferring the existence of that horizontal line. And that it's doing that because of the internal wiring in the visual cortex says, okay, if you've got a corner here and a corner here, there's likely going to be a line here, and there's like synaptic connectivity, which helps that happen. That's like a simple inference in neural dynamics process. We think that a very similar trick can be played by the frontal cortex. But now, we think a very similar trick can be played by our frontal cortex. But the representation that you're inferring over is not horizontal versus vertical lines, but it's like your future, right? Because the representations that we're working with in frontal cortex have a model of your future. And so what this kind of representation might be, it could take a representation of your present and an input saying your goal, where you want to go over here, and it could like fill in the rest of the future by inference in the same way that the visual cortex is filling in the rest of the visual scene by inference, right? And you can do that trick because the representation that you're working with is over the future rather than being over orientation of a visual bar, right? So that's what this model is suggesting can happen. And it definitely can happen in a model. It turns out that the other fun thing is if you want it to happen in the model, we also precisely know what synaptic connectivity rules you need. And it turns out to be quite an elegant set. The synaptic connectivity rules you need are a literal world model, as in, if I can get between two places in the outside world, I need those neurons to be connected in the consecutive layers of this model. So here, I can get from here to here because there's a bridge. That means that next has to be able to activate next plus one at this location, so the patterns of spatial activity in the inference machine can also activate this. Because if this one's active, that means I'm going to be here next, which makes it more likely that I'm going to be there at next plus one, right? And so if you just embed a literal world model into these connections, you can solve these funky planning problems. Okay, here's a pretty picture. I've played a trick that I wish I hadn't played, which is I've rotated my representation now. These are all neurons. But before I had position and position being on this axis and next and next and next plus one on this axis. And now I've got position being on this axis and next and next and next plus one on this axis. I'm sorry about that. I'm just going to show you that if you build this kind of model, it can solve quite sophisticated things like this complex maze task. So here you can see it finding the entire trajectory through space time and then activating on that trajectory. And it knows not only where you're going to go, but when you're going to be at each place, because the activity patterns were in different neurons ahead of you, right? And the activity is just resolving to the best solution of this problem, right? I'm doing inference over my future. We've done some analyses of these kinds of models, and I'm not going to show you those analyses, but I'm just going to tell you the results of these analyses, because I think the results of these analyses are quite similar to the results of PFC lesions. And maybe you guys are all going to disagree with me because you know PFC lesions better than I do. But I'm going to argue that the more dynamic and interactive the task is, the more PFC is needed for it. So here you don't need much PFC for a stimulus response task or a binary decision task. You actually, I think, we don't really know, but you probably don't need PFC for these simple spatial planning tasks as well. Hippocampus will do. There's all sorts of work up here in Edinburgh suggesting those kinds of things are true from Richard Morris. But you really do need PFC for these kinds of tasks when things start changing and different rewards happen at different places at different times. So like Paul Burgess down in London studies these wonderful tasks with PFC patients where he asked them to like go to Marks and Spencer and pick up three oranges and a shirt and meet Fred at 11 o'clock and be at the cinema at 12 and like all those, and they just totally fail on those kinds of tasks. I think because it requires them to hold a plan where different things are good at different times. And then once you get into theory of mind and game theory, that clearly has this interactive dynamic structure to it. So I can just tell you how these models do on these kinds of tasks. And the answer is the space-time attractor model that I just showed you is space and time. So it's because it's got a representation of the future, space-time. Those space-time attractor models can solve all of those tasks, but shouldn't be used to solve all of those tasks. Because these simple tasks at the bottom, anything can solve these tasks. And it's a waste of neurons having all those neurons that I showed you before. These kinds of tasks, you do need a bit more sophisticated. You have to have a world model, but you don't have to have an explicit model of the future. Something like the successor representation can solve these kinds of tasks. But these types of tasks up here, they kill things like successor representation. The moment rewards are changing in time, they kill those kinds of models, and you need a model with an explicit representation of time, like the space-time attractor. And so without showing you the data, I'm going to claim there's an interesting correlation between when you need these kinds of space-time models and when PFC lesions cause deficits in human tasks. Cool. Okay. The last thing I'm going to tell you is if you take versions, is it the last thing? Not quite. The second last thing I'm going to tell you is if you take versions of these complicated space-time tasks, like the ones Paul Burgess, and you just train a recurrent neural network on them, then it learns exactly a space-time attractor. It learns a representation of the future, and more than that, the connectivity weights that you see inside the recurrent neural network are exactly a world model. They're exactly predicted by. So this is the handcrafted one that Chris made out of his mind, and then this is the RNN trained one, and they're just exactly the same. That means, in my mind, that it's not a mad model, because like it's the best that you could do by backpropagation, learning on a recurrent dynamic system, right? And so, so it's not a mad model. Last thing I'm going to tell you is we don't know if it's true or not, this last part of the talk, but there's some fun things that you can do in mice to find out that would be hard to do in humans. And so this is what Sandra Reiner in the lab is trying to do now, which is to like measure using calcium imaging where all these cells are in this network. And then you can use these fun techniques now where you can go and stimulate like one neuron at a time or particular patterns of neurons. And then so we could maybe go and stimulate these green neuron, and then we see, okay, the ones we stimulated are active, but there are also some other ones that are a little bit active because there are connections, synaptic connections between these neurons. And then the question is, well, are these neurons the ones that we predict to be connected in these kinds of models? And so those are the kinds of things we're trying to ask in the lab, in the lab now. And that's some of the reasons I like being able to do stuff between humans and mice. I just wish I'd been able to show you some of the human data on the same task that we have, which is nearly ready now, but I can't. But I'll take questions about it, but I don't want to claim it's true yet. Okay, so that's what I've been saying. PFC as a model of the future, neural connections embed the task structure, and this might let you infer plans about the future. And you should treat those three things with very different levels of seriousness. As you, okay? And these are the people that did all the work. Thanks very much indeed.